So my chapter 10 vision outline started on brightness is created by the visual system. It starts with lateral inhibition, which is the phenomenon by which interconnected neurons inhibit their neighbors, producing contrast at the edges of region. So this occurs when neurons in a region, retinal cells, are interconnected either through their own axons or by means of interneurons, and each neuron tends to inhibit its neighbor. Our visual experience is not a simple reporting of the physical properties of light. Instead, our experience of light versus dark is created by the brain in response to many factors, including surrounding stimuli. The next major section is neural signals travel from the retina to several brain regions. So ganglion cells in each eye produce action potentials that are conducted along their axons to send visual information to the brain. These axons make up the optic nerve that brings information to the brain on each side, which eventually reach the visual cortex of the occipital lobe. There they have the optic chiasm, which is the point at which two optic nerves meet. Axons from the retina on one side of the nose, called the nasal hemiretina, cross over to the opposite side of the brain, while axons from the side of the head, which is the temporal hemiretina, project its axons to their own side. Having eyes cover overlapping sections of the visual fields help us to judge the distance of objects and by comparing the inputs from the two eyes. In prey species such as rabbits, natural selection favored broader visual fields that result from them having eyes positioned farther apart, so most of their optic nerve fibers cross the midline. The optic tract is the axons of the retinal ganglion cells after they have passed the optic chiasm. Most of them terminate in the lateral geniculate nuclei. Most axons of the optic tract in mammals terminate in the lateral geniculate nuclei, no matter what the species. The lateral geniculate nucleus is the part of the thalamus that receives information from the optic tract and sends it to its visual areas in the occipital cortex. Axons from the neurons in the LGN form optic radiations, which terminate in the primary visual cortex. Optic radiations are axons from the lateral geniculate nucleus that terminate in the primary visual cortex of the occipital lobe. The primary visual cortex, also known as V1, also called the striate cortex or area 17, is the area of the occipital lobe where the most visual information first arrives at the cortex. Some retinal ganglion cells send their optic tract axon to the superior colliculus instead. That's in the midbrain, and it helps them coordinate rapid movements of eyes toward a target and controls the size of the pupil. The extrastriate cortex is a visual cortex outside the primary striate visual cortex, and that processes things such as form, color, location, and movement. The next section is the retina projects to the brain a topographic fashion. The retina represents a two-dimensional map of visual space. This, of course, through the brain, it goes point-to-point correspondence between neighbors that are parts of visual space, and it maintains a map-like formation. Much of this is dedicated to the foveal area. Half of the human brain, V1, is dedicated to the fovea, and it makes for a great acuity of spatial discrimination in the central part of the visual field. If we know the site of injury and visual pathway, we can predict locations of a scotoma. A scotoma is a region of blindness caused by injury to the visual pathway or the brain. A person cannot perceive visual cues within a scotoma, but some may be possible. This is known as blind sight. The next section is box 10.2, which is eyes with lenses have evolved in several phyla. So evolution of eyes throughout the mammalian species and any of the chordates have followed the same steps. The first is concentrating light-sensitive cells. This means that they concentrated these light-sensitive cells into groups that served as photoreceptive organs, and they were able to respond to different stimuli that strike different parts of the body surface. The second was a clustering of these light receptors. They clustered at the bottom of pit-like or cup-like depressions in the skin and allowed for better discrimination among stimuli that come from different directions. The next part was a narrowing of the top of the cup, and that narrowed into a small aperture so that that can act like a pinhole camera, and the eye could focus well on specific things. The next was the closing of the opening with a transparent skin or filling the cup with a transparent substance. This covering was meant to protect the eye against entry of foreign substances and stopped injury to receptor cells or the blockage of vision. The last part was the forming of a lens, which was the thickening of the transparent skin by modification of the tissues of the eye. This adaptation improves the focusing of the eye while allowing apertures to be relatively large to let in more light. The only requirement for the beginning of this evolution appears to be the existence of light-sensitive cells. This is an example of convergent evolution in many phyla. This goes across octopi to mammals. All of these eyes share two genetic features. The first is that they use opsin pigments, and the second is that they develop these genes through the Pax family. The next major section is neurons at different levels of the visual system have very different receptive fields. A receptive field is the stimulus region of features that affects the activity of a cell in the sensory system. Photoreceptors excite some retinal neurons and inhibit some others. At resting potential, both rods and cone photoreceptors steadily release the synaptic transmitter glutamate. Light always hyperpolarizes the photoreceptors, causing them to release glutamate. Bipolar cells that receive glutamate differ depending on what kind of glutamate receptors they have. Glutamate depolarizes one group or bipolar cells, but the other will hyperpolarize another. These groups then are said to have different receptive fields. There are on-center bipolar cells, which are a retinal bipolar cell that are excited by light in the center of its receptive field. By turning on light, it becomes excited and receives less glutamate, and glutamate inhibits the on-center bipolar cells. The next is off-center bipolar cells, which is a retinal bipolar cell that is inhibited by light in the center of its receptive field. So turning off a light field excites the cell because it receives more glutamate, which depolarizes off-center bipolar cells. Bipolar cells also release glutamate, and glutamate will always depolarize ganglion cells. So there are on-center and off-center ganglion cells. On-center ganglion cells are a retinal ganglion cell that is activated when light is present at the center rather than the periphery of the cell's receptive field. This depolarizes the bipolar cell through on-centered ganglions. Off-center ganglion cells are a retinal ganglion cell that is activated when light is present in the periphery rather than on the center of the the cell's receptive field. Next section is neurons in the retina and the LGN have concentric receptive fields. So there's patterns of connections of photoreceptors to bipolar cells, and they're in different shapes of the receptive fields in the ganglion cells. Specifically, the receptive fields of ganglion cells are concentric. So photoreceptors in the center of those areas in the ring tend to have the opposite effect of those cells in the next circuit. Both bipolar cells and ganglion cells have two basic types of retinal receptive fields. The first is on-center off-surround, which refers to the concentric receptive field in which the center excites the cell while the surround inhibits it. The next is off-center on-surround, referring to the concentric receptive field in which the center inhibits the cell while the surround excites it. The primate LGN has 6 main layers and some smaller layers in between. These layers are bent like a knee. There's the parvocellular, which refers to a relatively small cell, and the 4 dorsal layers of the primate LGN are parvocellular. Magnocellular refers to the relatively large cells, and the 2 ventral layers of mammals are magnocellular. Neurons of the LGN have concentric receptive field, especially in magnocellular cells. They have concentric receptive fields that are large, receiving inputs from large ganglion cells, which receive their input from bipolar cells contacting many neighboring photoreceptors. These are not involved in color discrimination. Neurons of the parvocellular layer have relatively small receptive fields and can be traced back to cones. Neurons in the visual cortex have varied, complicated receptive fields. Neurons from the LGN send their axons into V1, but the spots of light that are effective stimuli for ganglion cells and LGN cells are not very effective for cortical cells. Visual cortical cells require more specific stimuli that are more elongated than those of the activated LGN cells. Most cells in the V1 respond best to lines or bars in particular positions at a particular orientation in the visual field. Some cortical cells also require movement of stimulus to make them respond. There are simple cortical cells, which are also called as bar detectors or edge detectors. And there is a cell in the visual cortex that responds best to an edge or a bar that has a particular width, as well as a particular orientation in the location of visual field. The complex cortical cells, which are a cell in the visual cortex that respond best to a bar of particular size and orientation anywhere within a particular area of the visual field. These respond to a bar of particular size and orientation anywhere. The more complex events are, they build up inputs from smaller ones. A simple cortical cell can be thought of as a receiving input from the row of LGN cells, and a complex cortical cell can be thought of as a receiving input from a row of simple cortical cells.